join us today. Okay, let's start, and you can learn a little about me and the class. Hopefully this will be fun. So I wanted to say a little bit about me. You'll learn more about me, is that I'm a professor here at Duke, and this actually is my 16th year at Duke, which is longer than I've been anywhere else. I'm actually professor at several departments, primary in computer science, but I have joint appointments in biochemistry, math, and chemistry. And students from most of those departments are also in my lab, which is a great thing about Duke. So I'm split between Trinity and the medical center, spanning those departments. And at other times, I've had appointments in engineering, although I don't know if I still have an appointment over there right now. Okay, so let's talk about kind of where this course lies in some sense. So really, this area of computational structural biology is really kind of between math and CS and sort of this area of chemistry, biology, and possibly applications to medicine. So let's explore that a little bit, and especially for those of you, those of you who are new to this, maybe new to some of you, but let me sort of phrase this a little bit. So when we consider, as you know, for the engineers in the audience, people coming to biology from a different background, I don't mean biomedical engineers, by the way, I know you know what I'm talking about, but is that we're going to be very interested in proteins in this class. So proteins are these linear polymers that are made up of these building blocks amino acids. And you can kind of think of the amino acid sort of as a graph here as having a kind of fixed part and a variable part. The fixed part is basically the same, except for cis trans-isomerization, and the variable parts are these different side chains, 20 naturally occurring proteinogenic amino acids. You can see different ones are shown here. We have a big phenylalanine, little guy here like this. So these give lots of different chemical properties and flexibility. Primary sequences like this, familiar to, say, genomics people. This folds up into regular secondary structure, things like alpha helices and beta strands, illustrated like this in the sequence, and then into these beautiful 3D shapes. And by the way, on the course website, there are some books I recommend to get up to speed, particularly Brandon and Tu's Introduction to Protein Structure, and also certain chapters of the famous textbook from Lubert Stryer at Stanford called Biochemistry. I highly recommend these. TAs may have others that they recommend. So here are two views. Here we see all of the atoms, including the hydrogens, and then these ribbon diagrams, which were invented by Jane Richardson, our wonderful collaborator and friend in biochemistry. To visualize this, this is actually a picture of a designed protein from our lab in collaboration with Len Spicer. And we're interested in these structures, particularly because these structures are designed to bind to antibodies, and the epitopes here are defined by certain HIV proteins. This is a chimeric protein that was designed to have certain dynamical properties. So it's a sort of example of this. So basically from a, if you will, from a engineering point of view, I want to appeal to everyone in the class. When you look at the cell, right, there are all these signals flying around and being transduced in various ways. And so that, we tend to think of that every now and then in engineering metaphors of what are the signals and what's happening here. But the fundamental thing to think about if you're coming from computer science or electrical engineering or another field is that, the cells are different from computers and electronics because there are no wires. Cells are wireless in some sense. And signals are transduced by a sort of diffusion process of small molecules, typically called ligands or called inhibitors, that diffuse to the properties and interact with these receptors or molecules like these big proteins. And they interact through a process of molecular recognition, which is mediated by the shape and the electrostatic and other properties of the molecule. Now, and this is why we're super interested in the physical properties, because that mediation to transduce the signal is based on these physical and chemical properties in some sense. So we're really obsessed with the shape of these structures because that's what determines whether the signal is actually transduced, namely whether a small molecule, an inhibitor, a drug, a hormone, whatever, is actually binding to these proteins. So that's kind of the setup for thinking about what we're thinking about here. Now, two of the big themes in this class are the kind of complementary views of this kind of biology. So basically the two, not inverses, but think go in different directions, are structure determination and structure-based design. So the first is, can you go from a gene all the way to determining its structure? And the second is, starting with a structure, can you redesign it to do something else? Here's an example that we'll talk about a little bit. You know, this is from my work, but we'll talk about it as a didactic example a little bit in these first lectures. Here we start with a gene from a non-ribosomal peptide synthetase. So here is basically a megaprotein that makes natural antibiotics, antifungals, siderophores, immunosuppressants. And there are various bacteria that make these as warfare against other things that want to consume or eat them or compete with them. And this particular module is going to adenylate a phenylalanine, phenylalanine being something like this kind of structure here, free floating. It's a chemical picture of the side chain of a phenylalanine amino acid. And adenylate, I mean, we think of adenylation in genomics as adding an A, but adenylation in biochemistry means basically the reaction where you take a phenylalanine plus ATP, and then you have a reaction in which the adenosine monophosphate is covalently bonded to the backbone of the amino acid to activate it for a subsequent reaction, and inorganic pyrophosphate is released. This is an enzyme catalyzed reaction, basically. So this is an important thing, and basically it allows you to incorporate the phenylalanine into the backbone of a peptide that is a very strong and a naturally occurring antibiotic. So these are great interest in pharmacology. What we can do with that, even if it's a big protein like this, this domain here, this genomic sequence, we can get data on this. This is from NMR. This is actually NMR of a 65 kilodalton protein, so rather large. And then from that data, this is a cluster. There are algorithms that interpret this data to determine the structure. Here's sort of the key thing about these biophysical techniques like NMR, crystallography, cryo-EM, is that these are not per se imaging techniques. They have as subroutines imaging techniques, but they're not imaging techniques. These are techniques in which the measurements you make are indirect, and therefore to determine structure or function or dynamics, you must make inferences. Let me reiterate that. In these techniques, the measurements are indirect, so you must make inferences. So immediately we're in the realm of thinking about computer science, mathematics, engineering, because we have to be able to make those inferences in the best possible way. But then when we can do that, we can reconstruct these structures. And they're related to a lot of techniques used in robotics, machine vision, image processing, and so forth. Once we have the structure, we can use that. Again, there's a computational process where we say, well, we liked it that it would adenylate, i.e. attach, adenosine monophosphate to a phenylalanine, but what else can we do with this as engineers? Well, there are two things you could do. You could try to take a protein and design into it an entirely novel function that it didn't have before. Like basically you could take a catalytically inactive protein and you could engineer in an active site in the machinery so that it would perform some reaction. Or you could start with a protein that performs some function on a native substrate and engineer it to have a different reaction. So here's one example. We're using the structure and of course, geometric physical reasoning and algorithms. We're able to switch the specificity of the enzyme by making site-directed mutations to the active site so that it no longer prefers to adenylate or act on the input of leucine, but rather wants a phenylalanine, but rather wants to work on leucine. So we switched its specificity for phenylalanine to leucine. So this is a case, you can think of this in computer science term. The type, this is a machine or a program, and the type of its input is phenylalanine. And by re-engineering it, making a few changes that were predicted computationally in silico, we changed the type of the input. So the input it wants now is leucine instead, and it will adenylate that. So basically two parts, determining or predicting structure, and then predicting changes, that is typically mutations that will change function. These are two themes that will interplay throughout. Why am I showing you a non-ribosomal peptide synthetase that's making an antibiotic? And of course, it's supposed to be about machine learning and COVID. Well, we need some basic tools. So the key question is, if we have techniques like this, how can they be applied to other systems to help us during the COVID pandemic? That's a fascinating question. Maybe some of you will like to think about that for your project. The second is, of course, that I haven't mentioned the role of machine learning here, that many of these things have sort of a module of machine learning built into them, but how can we do something better using these fantastic new tools for deep learning that are coming online and being used by labs all over the place. So there's a little mantra, if you will, for how we might think about some of these problems. Okay, feel free to interrupt anytime with questions. So what do we use from engineering, math, and computer science in this class? Like, you know, I wrote some things down here. So what things are used in these algorithms? Lots of combinatorics because we're counting things. Lots of tree search. We have tree searches. Best first search. We'll have a recitation that Paul and Farooq believed entirely devoted to A-star search, which is a beautiful AI technique that is used here. Graph algorithms. We'll talk about configuration space from CS230. You know, stream, set theory, functions. We'll talk about whether these algorithms actually are mathematically correct. In other words, are they given the right input? Will they compute the right answer? And of course, probability comes up here all the time, and Bayesian approaches. So lots of different things get used here. These are just from an undergraduate class and more advanced techniques as well. I want to mention the textbook again. So this actually came out of an earlier version of this class. Again, 50 chapters on different topics, trying to look at key papers and results and summarize them really briefly. Did I succeed? Some of the chapters are longer than I'd like, but many of them are a few pages and they summarize the key points. So the idea is that each chapter is designed as sort of a little koan. It's hard to understand, but once you do, you'll understand the topic. So looking at the original paper together with the textbook will be really helpful, and we'll do that at least in the beginning. All right. How about reading and presenting papers? Like what kind of things will we be talking about? So we're going to start off with this beautiful paper from Steve Mayo's group, classic paper at this point, where he designed a zinc finger. So this is really interesting. We'll talk about it before, but this is a case where there's a naturally occurring protein that looks like this, and he wanted to do essentially what I would call a beautiful engineering hack. He was like, I know this evolved naturally, and it chelates zinc in a tetrahedral configuration like this. It does all these wonderful things. Can I design a de novo sequence, a totally different protein sequence that folds up to the identical shape? I don't care what it does with zinc, it just should have the same shape. And he asked the question, would it be the same as the wild type sequence? In fact, it was wildly different and much more stable. So this is a fantastic exercise and inspired many of us to go into molecular design. So we're going to read that, and it's going to be the basis of some things we'll think about. Again, this is all on the course webpage. What about other papers? I promised there'd be some things on SARS-CoV-2. So the design algorithms that Steve Mayo pioneered now are used throughout the biochemical and pharmaceutical world, and lots of different things have been done. So here's a paper. We read this last year. I think we'll read different papers this year, but to give you an idea, here's using these design algorithms to design very tightly binding inhibitors that bind to the spike protein of SARS-CoV-2. These are small proteins, if you will, biologics. It's a very interesting paper. I want to pause for a minute to introduce our second TA, Paul Saberwal. Paul, do you want to come on and introduce yourself and maybe say a fun fact about yourself?
Sure. Hi, I'm Paul. I'm TAing this class after having taken it last year, as well as I currently work in Professor Donald's lab. And unfortunately, I have other classes from the 1:45 to 4:45 block, so I won't usually be able to make our lecture, but I will see you guys in recitation and office hours. And particularly, I'm gonna try to help you guys through using Pymol and Osprey if you want to use it and all that kind of stuff, having taken this class before. So I'm excited for the class. Fun fact, I've been chased by a buffalo once. And yeah, looking forward to the semester.
That's great. Paul, I sorry to get your picture up there, but people will meet you soon enough. I think I have myself spotlighted, which is the issue. So that was great. An interesting thing was there was a certain convergence among the fun facts. I think some people that went first influenced other people as to what they would say. So by and large went in the directions of travel and music. But yours was totally unbiased, and we get buffalo chasing instead. So interesting sociological experiment there. So Paul, thanks for coming, and I think you all will enjoy working with both Paul and Farouk very much. If there are no questions, I'll go back to the presentation. All right, as I mentioned, this is an example of using silico design to predict things that would bind to the spike protein of SARS-CoV-2 and therefore prevent its attachment to the ACE2 receptor and therefore infection. So very interesting. So let me give you another example. This is from my own work. So basically one of the things we've learned about SARS-CoV-2 is that the antibodies and T cells are super important to how we defend against this. So what you may not know is that the design of the protein for both the Pfizer and the Moderna vaccines was essentially done by Jason McClellan, who at the time was at Austin, UT Austin, but had done various training at the Vaccine Research Center at NIAID, NIH. And he'd worked on HIV, but he'd also worked on RSV, respiratory syncytial virus, and designed with his group a vaccine that's now coming online to protect children against RSV. And he found a trick by which he was able to lock the proteins to which you want the antibodies to bind into a particular immunogenic conformation. It's quite flexible by using a special trick. And when he and his postdoc at Austin saw the sequence to the spike protein of SARS-CoV-2 wild type, they immediately went to cryo-EM, which he's an expert at, and they solved the structure at high resolution, published that in Science. 2 weeks later, they had designed the protein that's used in these vaccines. There's subsequent work to be done. You have to cap the RNA, you have to substitute uridine with pseudouridine, you have to optimize the RNA for very quick transcription, and those are basically done by really good work based on RNA labs. But basically, this was a protein design problem. So the other bet that I'm having here, value proposition, is that the work that we do as scientists on antibodies from other viruses is going to be super important to further advances in thinking about therapeutics, antibodies, vaccines against SARS-CoV-2. So I want to tell you some about that. Let's go back to this example here. So motivation is, antibodies bind to the envelope or outer part of HIV and neutralize the virus. So what do those regions actually look like, right? This is the goal of structural biology, so you can design better therapeutics. And again, I talked about this example, which tries to basically dynamically capture some of those regions, structure that we solved by NMR. So I want to tell you a little more about why I think this is really interesting and important. And I think the application to novel viral targets will be self-evident, or at least by the end of the class. So let me give you a very personal digression or odyssey through the virology world and how these algorithms, I think, can really help. Again, I'm not going to assign these papers because I don't want this just to be about my work, but I want to tell you where I'm coming from, and maybe you'll find it interesting. So these are some of the things that my lab works on, and they're basically 2 things I want to tell you about that I think are relevant to our investigations in the class. And the first have to do with resistance. Can we predict resistance to therapeutics or to antibodies before deployment? And the second, of course, is can we design antibodies and can we design vaccines that are better? So I want to not tell you about all of our papers and results, but I want to instead tell you about just a couple of these as a little personal odyssey, if you will. Finally, one thing I do want to mention here is that it is interesting when you evaluate these papers, you want to evaluate, do the actual molecules work, right? That's pretty interesting. And then you also want to ask questions like, how efficient were the algorithms? But you also might want to ask, how many constructs did you have to try in the wet lab? So basically, one interesting thing about Steve Mayo's original paper that I talked about is they just tried one thing. They basically predicted a sequence, and then they made that sequence, it folded up the right way, they verified that experimentally, and then they published it. So this might seem anecdotal, like just one sequence, but actually it's a triumph. It's what you want the computation to do. It gives you the answer and you try it. So lately, a lot of the success of computational protein design has been coupling these computational screening techniques with high throughput experiments. which is a great idea, of course, if you have access to those experiments. But not all of you, as you go on in your careers, are going to have access to those. You might be starting a lab where you have 5 or 6 grad students and not 60 or 80 or 100 graduate students, right? And in that case, maybe you can't try zillions of constructs. And there are many papers where people are partnering these algorithms with techniques where they're screening 100,000, 200,000, or even 10,000 constructs. So here in red, I put the number of constructs that were tried in order to get these results. And I think it's not 1 all the time, but it is pretty low. So I think that's something we can evaluate as well, is what's your experimental burden? And how much can the algorithms alleviate the guesswork that has to go on in the lab in order to predict these sequences that will have these properties? Okay, so let's go on with this. Again, what are these antibodies? What are the reasons the antibodies bind to look like, and how can we exploit that? So, and what could you learn from the textbook and this class? So when I was writing this textbook, the editors said, well, what about a cover for your textbook? And I proposed this cover here, Protein Redesign for Dummies. And I thought it was pretty funny. The editors, the MIT Press did not like that at all, and they came up with a much more sedate cover instead. But I think ultimately, in some sense, I'm not at all saying you all are dummies, just riffing on the style of book here. I think that in some sense, we are getting in the first few lectures a little crash course on how do you do protein design. I hope you found that amusing. Let me mention that generally, if I'm speaking live, there's a little bit of chuckling at that point. One exception was once when I went to the University of Maryland, which is actually a great place, all sorts of cool stuff going on. And I gave this talk, and when I showed a slide like this, no one laughed. And but it was a great visit. I met with the deans, and I went out to dinner with people and talked to the students. They were very complimentary and friendly. And I found out why, is that there's a guy on the faculty there that actually writes these books, right? That's his thing. And so when they saw this, they didn't think Donald made a joke. They thought Donald wrote a book, so what? So anyhow, you have to be careful what you say. But he wasn't offended. He thought it was pretty fun. Okay. So how do we do protein design? Like, let me give you a quick sketch of really what goes on here. This is very much simplified. And I'd say this paradigm works for a bunch of things. Protein interface designs work for cystic fibrosis, HIV in some sense. So basically you have a starting structure. And of course, we'll talk about how do you actually compute those structures or predict them. You have a starting structure, and there's interactions between 2 proteins here, schematically illustrated as the different side chains. So the thing is, if we want to redesign one half of this to like bind more tightly or bind more specifically or bind to something else, we take away the side chains, then we try to put them back. And we have a library called Rotamer library that stands for rotational isomer, right? These are all the same chemical formula, but they have different shapes in some sense. And basically the shape determines the amino acid. The main purpose of this slide is that they search over all the different possible amino acids and the simultaneous search over all possible conformations or shapes of these, which are parameterized by the angles going up the side here, the chi angles, and of course the backbone. This is an enormous space, so we need better algorithms. So the only point of this animation in some sense is that to search this brute force would take forever. So we need fancy, faster algorithms. That's where computer science comes in, to search over the library of things you can stick on here to get a different effect. And of course, this is what you'll be reading about for next week. So of course, you can subsample these, like you could say, well, those are very far apart in shape space. You could subsample these and get things like this. You have tyrosine interacting with the leucine over here like this. But the basic point is that you have lots of these. Here's the combinatorics. You have the product of the number of rotamers at position 1, position 2, position 3. This is an exponential number in the number of simultaneously mutatable positions. So just because the search space is exponentially large does not mean the algorithms have to be exponential, as it turns out. So we had used algorithms here that are driven from engineering and computer science and computer vision to do a better job with this. That's sort of the key way of thinking about this. So If we can search this quickly, we want to rank these based on a computed energy. Here we have a pairwise decomposable energy function, or if you're in computer science, this is a Markov random field. So we have one body and two body energies. I believe that Hunter will be telling us about energies that don't have to be one body and two body, in particular bringing in quantum mechanics. But basically the idea is that the top conformations and sequences are ranked by this in silico energy function, and the minimum, which is called the map estimation in computer science, has the jargon name GMEC, Global Minimum Energy Conformation. We'd like to compute this. So most algorithms are trying to estimate this, which is the lowest energy sequence and lowest energy structure. So an enormous combinatorial search. There we've defined the search problem relative to an energy function searching over this space. So basically we're trying to search for the red things that minimize this function. It's simultaneously a discrete search over the shapes and a continuous search over the discrete search over the sequences and a continuous search over the shapes. Okay, so I'll just tell you how we do it in some sense. There are all these different parts of Osprey in some sense, right? There's these, this input model has the input structure, which is 3D. There's the rotamers, which are these red things you can put on. There's the energy function. Sort of talked about those. And then there's some kind of pruning here based on flexibility. So basically the idea of this is we want to prune things based on using discrete or continuous flexibility. One of the things we can do here is we can use algorithms from computer science, like dynamic A star or so forth. And what we can do is we can actually enumerate the conformations and sequences, starting with the lowest energy one. Lowest energy is the best one in physics for this, without any gaps in an order of energy. So this is something we want to do just because we care about models. We want the best solutions. But one of the things that happens here is that this allows us to actually compute information about binding. So it used to be maybe 15 years ago when I talked about this kind of material that I had to be very cautious around computer scientists because they hadn't seen partition functions, whereas the people in physical chemistry and biochemistry and engineering had. But now, of course, partition functions are everywhere in machine learning and data science. And so you might have seen them, and if you don't, they'll be super useful there. So basically the idea is that the binding affinity of one molecule, say an inhibitor for a protein, is defined in the following way by essentially counting the number of conformations and weighting them Boltzmann fashion by their energies. Here's the Boltzmann weighting of the energies. We have these enormous sums here with enormous scientific computation problem trying to estimate not only this enormous sum over conformation space, but also its quotient. So the trick turns out to be that if you are a computer scientist approaching this problem and you wanted an algorithm that not only gives you the best answer, but that enumerates the conformations in order of energy without gaps, then what you can notice is that you can bound the tails of these distributions and you can turn off you can stop the computation when it's accurate enough. So basically you have this enormous stream of conformations being enumerated for you in order of energy. And you're going to start with the lowest one, and you're going to add these up and keep a running sum. You can bound the tail and actually get provably good approximations to this binding. And we'll cover topics like this later on. Was there a question? Okay, so there are various tricks here. It turns out that if you take it gives you greater functionality because instead of just designing to a single state, you can look at binding as well. So another technique that is useful here is looking at these rotamers not only as discrete, but as continuous entities. So here we see an interaction here. This is from interleukin 14, which is a kind of immunosuppressant system. And what we've done here is we're just slightly rotating these side chains. And what you can see here are probe dots from the Richardsons, and when they go red, it's a clash, and when it's green and blue, it's a very good interaction. So slight changes in the energies here can make a huge difference. And that's why there's both a discrete and a continuous component to modeling these conformation energies. Okay, so we're going to talk about the interplay of continuous and discrete. It also is a similar interplay than we see in other fields like robotics and machine vision. Of course, anything that interacts with the real world is interacting with a continuous domain in some sense. And the key thing is how can we come up with a discrete computation in that case that still can give us provably good answers. but I wanted you to see this picture of kind of the interaction between small continuous changes versus really discrete choices, such as changing one of these guys here to be a big ring like a tryptophan or a phenylalanine or something like that. Okay, so basically, let me give you a little hint of how these algorithms can actually do something in some sense. So if we pass to the configuration space of these systems, what does that look like in some sense? So here we have a particular amino acid right here. It's isoleucine. And basically the degrees of freedom here are two rotatable dihedral angles, chi1 and chi2. So it's kind of like a two-link robot arm, and we can graph chi1 here and chi2 here. These wrap around. This actually forms a two-dimensional torus because 0 and 360 degrees are the same, but we can kind of visualize it as a game of Pong or as the video game where we can visualize this as a plane like this. And basically the idea is that if you just discretize and look at the low energy regions, your representation are these points like this. And of course, slight variations in these points lead to very different energies. So here's a situation where we have a bunch of side chains, maybe in an active site, not a realistic case, but a theoretical case. And we get infinite energies because we have these enormous clashes between essentially the nuclei, not the electron clouds here. However, a slight minimization here, right, local minimization is going to get you beautiful energies instead. So there's several ways of kind of transforming this discrete problem into a mixed continuous and discrete problem. Let me give you ways of thinking about this that relate to different parts of engineering and computer science. So the first is that if you're a biophysicist and you just design with these discrete points, you'll rule out this sequence because it has enormously high energies, which is bad. And that's a wrong thing to do, because if you made this sequence and then just slightly minimize these things, you get beautiful energies. So in some sense, you want to modify the algorithm to be what I'll call minimization aware. In other words, you can start with this conformation, but then imagine you're going to predict the effect of minimization, which will be done ex post facto when you have the entire structure. So this involves replacing this geometric model of 3 points in this two-dimensional space with 3 regions like this, maybe plus or minus 9 degrees. And this corresponds much better to the biophysicist's notion of what a rotamer really is, namely a region of conformation space capturing an energy well. So in other words, if you were to do a semi-smart thing, design with these energies and then minimize your structures a little bit at the end, you'd miss this beautiful solution because it didn't have good initial energies. However, if you just treat these points as specifying regions and then ask for energy bounds over these regions, the maximum and minimum, you can then design for these things being plausible, and you can predict the results of the ex post facto minimization you'll be doing. This is actually deeply connected with something that happens in computer architecture called data speculation or execution speculation. You're basically speculating on the results of an execution that hasn't happened yet. But from our point of view, we can think of this as going to something like interval arithmetic. In other words, we have a bunch of calculations we're doing and doing these sums of Boltzmann-weighted energies, that's e to the energy. And instead of using that, instead we're going to have minimum and maximum bounds instead and calculate things. So doing this, we can get far more accurate predictions. So basically, 2 of the key features of getting more accurate predictions are using this notion of blending continuous and rigid or discrete computations, and also using these conformational sets or ensembles in some sense. And going back to the ensembles, if you take any kind of biophysics or biochemistry class, you know that you have to look at more than a single structure. So traditional protein design just looks at this single structure, but instead we know that proteins actually exist as a thermodynamic ensemble. There's more than just the lowest energy structure for binding. And in fact, you have these partition functions. Instead, this is the energy of the bound complex, the energy of the 2 components when they're unbound instead. Think of these as a stream of energies that are weighted by the exponential or Boltzmann function here. And this actually turns out to be exactly the same quantity as if you measured the concentration of the complex divided by the concentrations of the 2 components here. And it's also assuming what's called the ergodic hypothesis that says the time and the ensemble average are the same. It's the same as doing a molecular dynamics trajectory through time and averaging the state over time for a sufficiently long trajectory. So time, ensemble, and concentration averages all give you the same binding constant, which is how tightly does something bind to something else. And this, of course, is of enormous consequence when you read these papers that are preprints or articles in the New York Times, and they say, look, this antibody we thought was really good, it's a monoclonal antibody, does not bind as tightly to the S protein. Well, these are things that you can hopefully calculate and you can redesign a better antibody. But back to our problem here. The key insight here is an algorithm called K-star. Again, it's like series or streams and CS230. You use ensembles of structures rather than just a single point at the bottom of the energy well. And you basically look at the essentially the energy weighted partition function for the complex divided by the partition functions for the ligand by itself and the protein by itself. So that's sort of a key trick that's extremely useful, but difficult to calculate in the sense you have to think hard, and so therefore you need sophisticated algorithms. Therefore, I'd argue you need a class like this in order to be able to do that. Okay, so finally, let me just mention a couple of points from the point of view of maybe the kind of empirical side of what we're talking about. You know, as a mathematician, it's very easy for me to kind of motivate or talk about the beauties of a provable algorithm that's guaranteed to give you the best solutions during the model. And we believe in things like that in engineering and computer science. But let me just mention that as an experimentalist, there's something really useful about these kind of algorithms. So if you use some kind of heuristic unprovable algorithm, and here are three genetic algorithms, Monte Carlo, self-consistent mean field. I mean, some of these have some guarantees in limited cases, but these will give you some solutions, and they will be relatively good sometimes, but you're definitely going to miss things. And if you have an algorithm that provably enumerates the solutions in order, starting with the best one as ranked by your scoring or energy function, I will tell you that as an experimentalist, and I have an experimental lab, and there are even people from that lab in this room, that these solutions that are missed by these heuristic algorithms look really interesting, and I want to try them. And so that's, I think, a really strong reason for looking at these kind of algorithms and seeing what we can get. So not only might they be better, but they look really interesting and diverse. Okay, we'll come back to this point in a way. So let's now get back to some applications. I mentioned resistance to motivate, my interest in these problems. It's not a paper that we'll read, but we'll talk about it. So this is about resistance to antibiotics. Now, those are different from antivirals in some ways, but otherwise sort of similar. I just want to talk about what the goal is here, which is to predict ahead of time what the pathogen or microbe is going to do. And by ahead of time, I mean in silico. Can you do it on the computer? Let me give you a motivating example, which is from the antiviral space. So I'm sure you all know about Tamiflu, oseltamivir. So this is a relatively new drug. When I was growing up, we didn't have antivirals against influenza. And I actually used to forget about it when my kids would get the flu when they were little. And my wife would remind me we should get Tamiflu, or the doctor would say that, and they would take it and it would help, I think. Here's the package insert for oseltamivir or Tamiflu. This goes to the patient. This is not just for the doctors. It's all about resistance. And look at what happened here. This one is telling you that at residue 292 on neuraminidase, which is an important flu surface protein, an arginine gets changed to a lysine. And here we have, a histidine being changed to a tyrosine and so forth. Now, how do we know about these mutations? And these are mutations that make the flu resistant to Tamiflu, right? So how do we know about this? This is all post-marketing. This is all after phase 3 and even a phase 4 follow-up from reports. So what happens is someone goes into the physician and they say, "Doc, I'm feeling terrible." And they happen to be part of a study, so they swab him in addition to giving other medicines, mucus, sputum, and so forth. And then they sequence the virus, right? They don't usually look at host factors, they sequence the virus. And then they do a statistical association to determine that these are the mutations in the virus that are statistically associated with poor outcomes resistance to Tamiflu. And then drug design has to start again de novo from in vitro characterization of proteins with these residues mutated, followed by animals, followed by humans, and so forth before it goes up. So the idea is, what if we could predict this ahead of time? It's a fascinating idea and very important, I think, for our SARS-CoV-2 focus in this class. So I wanted to tell you a little bit of what we know from other domains, and you can view this in some sense as a manifesto for can we apply them here. So let's talk about resistance for antibiotics, incredibly important problem. So resistance has emerged really quickly to all these antibiotics, right? And the reason it took a long time for some of these like vancomycin is that it was reserved or withheld by physicians for many, many years before being used. That is not reserved from the entire population. It was only given to certain patients in really dire circumstances. Basically, resistance emerges really quickly. So one thing my group did is we were really interested in looking at a particular target where we thought resistance would emerge and seeing if we could predict in silico on computer using protein design the resistance mutations that would evolve when you treated with this. So we looked at dihydrofolate reductase. So this is an enzyme that reduces dihydrofolate. So how to put that in context if you're not a biologist. So, pregnant women take folic acid, right? Why is that? Spina bifida, that's basically trying to enrich the amount of dihydrofolate. It's a very important key enzyme in both us and in pathogens like Staph aureus here. In this case, methicillin-resistant Staph aureus. It's a very key enzyme. It's important because it makes thymidine. Basically, it's part of the sole de novo biosynthetic pathway that makes the T in the genetic code. So if you can't make the T, and there are only 4 bases, right? There's G, A, C, T, right? You can't make the T, then the cell dies in stage S1. So you want to design a specific drug that binds to and disables dihydrofolate reductase. Again, the sole de novo biosynthetic pathway for T in the genetic code in the bug, but not in humans, or at least not as much in humans. Then you have a cofactor, which is NADPH. So we have inhibitors for dihydrofolate reductase like trimethoprim. And if you're ever taken Bactrim, like I hurt my foot a few while back, Bactrim is a combination of trimethoprim with sulfamethanol, right? So it's a sulfa drug combined with it, targeting 2 enzymes. Resistance emerges super quick to this, but it's still a useful drug. So my medicinal chemistry collaborators have been interested in building other molecules that can work. So this is a folate analog. It's one that basically competes with folate, binds more tightly. So, and here's how folate binds. So this is a new drug. It's called Compound 1. It has this interesting propargyl link here. So we wanted to try this out. It worked really well in rabbits. It worked well in vitro, worked well in structures. But we know that when we treat with this, we know that resistance might emerge, and it might be different. So this is different from the standard bioinformatic analysis where you look up a bunch of sequences and say, well, this happened before, so maybe it'll happen again. This is new chemical matter. And so it's possible that the resistance mutations could be different. So can we predict that on the computer? So here's sort of our approach. It's kind of a paradigm that I think is really interesting and relevant lots of places. So here's the key thing. And basically, if you get only one technical takeaway from this little discussion, is what I'd like you to think about. It'll come up again and again. So we want to reduce the problem of computing resistance to a problem of protein design because that's what we're good at. So I would phrase this as a hypothesis. The hypothesis is that nature, siding with the bug, is trying to solve a protein design problem, a really difficult problem that has 2 competing objectives that have to be reconciled. So the first is to survive against your drug. Nature is trying to redesign dihydrofolate reductase, or DHFR, to have different side chains, mutations to the active site that prevent binding of the new drug, right? Now, simultaneously, but perhaps with a different shape or conformational ensemble, those same mutations should maintain or strengthen binding to the natural substrate. So the first part is called negative design. Can we design the protein to not bind something? And then that same sequence, that discrete choice, but with a different continuous shape, possibly, should maintain or preserve or even enhance binding to the native substrate, the catalytic activities. That's positive design. Negative design as a concept was invented by Dave and Jane Richardson, by the way, here at Duke. And it's one of the things that these algorithms are good at. So in other words, I want a set of mutations to the active site, which is the place where it binds, right? And I want those mutations to not ablate, but hopefully maintain or even improve binding to the native substrate. And I want them to disrupt binding of the inhibitor. So we did that. We basically ran two different protein designs and combined them in negative and positive design, and then looked at the results and performed various analyses. So what do we get out of that? Let me show you some experimental results. So first of all, one kind of cool thing is because algorithms like Osprey are provable and they enumerate things starting with the best one, we get a ranked list of all the mutations. So there's no gap here. There's no incomplete optimization. We get these ranked in order of how likely they will arise, right? That's pretty neat. We restricted these to SNPs or single nucleotide polymorphisms because we thought there was more likely to arise in actual selection experiments. So these are what the algorithm says the protein will evolve to in order to evade our inhibitor. So then what do? Well, first of all, we do experiments to characterize this in vitro in purified components, right? So what we do is we measure enzyme activity. So some of you are computer scientists, and we have order notation to talk about the efficiency of algorithms. Biochemists have a measure of the efficiency of enzymes. It's kcat over km. Kcat is essentially the speed of the enzyme at doing catalysis, and km is the velocity of the reaction at half maximal concentration. So kind of bang for the buck in some sense. It's like, how quickly do you turn over the reaction given how tightly you have to bind and how much energy you have to use? And then you could also have ki, which is the inhibitory constant. It's how much do you outcompete how much do you outcompete the inhibitor? So basically, if you are interested in a better enzyme for the bug, you don't want a big drop in catalytic activity, right? And you do want a big drop in how tightly you bind the inhibitor. And that's what happened here in this graph. So notice in this graph, we have 4 different mutations here. These are the ones that are ranked here, 1, 2, 3, and 4 like this. And notice what happens here is that these bind to the inhibitor. The inhibitor binds to the Staph aureus target up to 100-fold less well, right? And the drop in catalytic activity is kind of minimal. So that's in vitro, right? Bug hasn't done this, but if we make the enzyme, it works that way. Then we did, and of course, one kind of cool thing is that you get a structural model out. So you get these detailed models of what the shifts here, and you can verify that structurally by solving a structure later. We then did resistant selection experiments. These are biochemists call these in vivo, cell biologists call them in vitro. These are selection experiments in a dish to see if we can actually take a colony and select for resistant versions. And here we measure fitness, which is essentially doubling time versus gaining resistance. That's change in minimum inhibitory concentration, or MIC. And you can see that this is also very interesting. The mutation that we predicted does emerge, but in concert with a previously known mutation of phenylalanine to tyrosine mutation to position 98. This could have been known from bioinformatic analyses. We knew about it. So this shows you really have to combine both. You have to combine the sequence-based bioinformatic analysis of what has happened in the past with new chemistry, new predictions to determine really what happens, which is a combination of these things. At any rate, the combination mutation of these things basically is up to 200-fold more resistance and almost no loss in fitness. And then we also solved the structure, which is, interesting. We were able to solve the structure of this and see the differences here. And we were able to look at whether these mutants actually occurred. So one pretty interesting thing about this story here that I'll just mention is that, as we talk about it, is that, I didn't mean to do that. I'm really sorry. To skip around like that, I must have done something crazy. The thing I wanted to mention is that subsequent to this, we actually found that in addition to appearing, so here we have in vitro purified components. On the right, we have what happened in cell experiments, but subsequently this mutation arose actually in hospital patients in Connecticut. So, pretty convincing that these algorithms can do something very interesting to predict resistance ahead of time. Then hopefully you can design better inhibitors. Okay, I was going to show you this slide. Here's the example of what happened in hospitals. So basically, in short, of the predictions that were made out of millions of possible mutations, basically the blue ones worked in vitro and the green ones arose actually in hospital patients. So pretty useful predictions to think about these things. Okay, so we think that these kind of algorithms, which we'll learn about more in this class, could be really useful at a variety of different targets, including, of course, potentially viruses like SARS-CoV-2. And again, maybe in the future, instead of having things like this, which are these package inserts based on post-marketing, we can imagine a world in which this was predicted on the computer ahead of time, and then we iterated drug design to try to design something that was less reactive and more proactive, basically looked ahead the same way our chess player was looking ahead against the bug. Okay. All right. Let me tell you one more story, and then I'll talk more about the papers we'll read. So one other case, I want to talk about virology again. So HIV is different from SARS-CoV-2, but there are some tools that I think could be really interesting here. So how does HIV attack the cell? So it undergoes all these conformational rearrangements, then it attaches to a receptor that's called CD4, and then it attaches subsequently to a co-receptor, then it invades the cell. There's fusion of the cell membranes. So of great interest to the medical and HIV community is, could we design sort of an inhibitor or something that would block this attachment? Can we inhibit binding to CD4? Right. So one of the things that we got interested in was antibodies that did this. So this work was done by a PhD student from my lab. After he graduated, he went to the Vaccine Research Center, and he, with a large team there, managed to find a new antibody that actually had this effect of blocking HIV infection. This is called VRC01, Vaccine Research Center. And when this happened, so the best antibody previously was this antibody B12, which is pretty famous. And this is the fraction of circulating strains that it neutralized, right? So VRC01 neutralized like 91% of the strains. So it's really interesting structure, really interesting antibody that's naturally produced. And the way this was found is, at the time, it was known there were so-called long-term non-progressors. So these are people that were infected with HIV, they're not on antiretrovirals, and they're not sick, and they're holding off the infection, keeping their viral loads down for a long period of time. So they fished around in their blood for basically looking for both antibodies and mature memory B cells that were binding to a stabilized form of what's called GP120, the envelope protein on the surface of HIV. And they pulled down both antibodies and these mature memory B cells, which are factories, and they sequenced the B cells, and they found these antibodies. At the time, there were only 4 people in the world. There were 2 from Africa, 1 from Europe, 1 from the United States that could make these. Now we know that between 10 and 15% of people infected with HIV can make these, but they're very hard to make, and these were not detected in animal studies like rabbits and monkeys because they don't have the right genomic chains to make these antibodies. Only humans and some other creatures can do this. So this is really promising for looking at an antibody therapy and possibly a vaccine for HIV around 2010. One thing they noticed immediately is that when you start treating at a therapeutic titer, which is more like 1 milligram per milliliter, as opposed to 50 mg per mil, the strains neutralized goes way down to about 3/4. And they were certain that if we do this, that you would be selecting for resistance, right? You're selecting for those highly resistant tier 2 viruses. And so they asked us, can we optimize this antibody, come up with a new antibody that has better properties at a lower titer, more potency, more breadth. And the project is using computational protein design. So really interesting work here done by Evelyn and his community. So basically, how do these antibodies pull down? Well, that's a whole story in and of itself. But basically, that's also a combination of positive and negative design. So you have these probes which are bait proteins. So you want them to bind to certain things and not other things. So basically, the positive design is to design the probes so that they bind to the right kind of antibodies and not the wrong kind of antibodies. So in some sense, you want the designs to be ranked up here. So they're very high on one scale and very low on the other. So again, you're designing probes that you're going to put into human sera that are going to try to pull down or bind to constructs that are the right kind of antibodies, right? So again, the key theme here is combining positive and negative design is one of the most important things that protein design can do. So that's kind of how the probes were developed. Let's go on to some results here. So using these better probes, they came up with this beautiful antibody that's slightly different from VRC07 here. And then we use Osprey to redesign the interface to make it broader and more potent. So here's the interface. The picture is like the one I showed you, but in more detail. We're trying to optimize or change the residues that are on the surface here. Here we have the heavy chain and the light chain. We're trying to basically change these side chains that would poke out here to bind more tightly to the HIV target and also more broadly against different strains here. And coming up with this, there are a whole bunch of mutations suggested. Like here's one where we mutated an isoleucine to an arginine at position 30 in the antibody. And you can visualize these as a particular one. And in silico, that is on the computer, this is predicted to have better interactions and bind more tightly to the target. So after a long process of looking at a lot of these different possible substitutions, these went into testing. So for example, this one here, you can see that there's an improvement. Again, IC50 is the inhibitory concentration at 50%. So basically, how much concentration do you need to inhibit half of the virus? And you can see an improvement here, which is kind of remarkable given this is heavily optimized antibody by the immune system. So we can find improvements here in silico that are predicted, that actually work in the lab, which is pretty exciting, 1.5 fold improvement, which is exciting. And these things can be combined. We'll also talk about some of the experiments that are used to get this kind of neutralization data. So this particular experiment was actually designed by David Montefiore here at Duke in the Duke Human Vaccine Institute. It's a really clever experiment coupled with these structural studies. So I just want to tell you about it so we can interpret this data on neutralization. This is very similar to neutralization data you'll see in the coronavirus studies as well. So it's worth thinking about. So let me go into experimental land for a little bit rather than computational land. So how do you measure in vitro, in cells, whether you have good antibodies or good therapeutics? It's, do you grow live virus? So HIV and COVID are both RNA viruses, right? So they have RNA as their genome and they're going to reproduce and they're going to do their thing to infect. So this particular system here, first what they do is they make a pseudovirus, which has a DNA genome instead. So they reverse transcribe the viral genome to DNA. And they break it into two parts, different plasmids. And one has the envelope proteins and one doesn't have the envelope proteins. Envelope proteins are here. This is all the rest of the stuff HIV needs. The reason they do this is that they want to have different envelope proteins for different strains. So in a 96-well assay, they vary this part between the different strains, keep this part constant. And then you get this, you transfect these into 293 cells here, and then basically this is going to produce virions, produce viruses, but the viruses can infect, but they can't reproduce because they don't have the machinery to do this because they don't have RNA. So they can just infect once. So you're not looking at amplification, you're just looking at basically do they infect. So you get these pseudoviruses, and then how do you measure whether they infect? Well, here we've engineered this cell here to have a basically a luciferase reporter assay. Luciferase is a protein from firefly that's going to glow when it senses HIV viral TET, which is an important virulence factor in HIV in the genome. So basically, when these things infect, they can only infect once. These things fluoresce. So you've got a certain fluorescence measure for whether they're infecting. If you then have, say, an antibody you've designed and you want to test this, it reduces the amount of infection. You get a quantitative measure for how little these things infect. So really beautiful assay that can be done in a pretty high throughput way. And that's how this data was obtained on, an improvement here from this particular substitution. But the exciting thing about this kind of work is when you can design better antibodies, obviously of great importance to all virology, is that you can characterize these things in various ways. So for example, here is a graph in the upper left where we're looking at potency on this axis versus breadth. This is fraction of the strain neutralized. So here B12, which isn't very good, things move up and to the left when you get better and better natural antibodies. These are natural antibodies discovered over time and they're getting more potent and broader. So we then can compare those with designed antibodies. Here's a designed antibody. Here you see the improvement here from basically VRC01 to VRC07 to the designed antibody here that has a number of substitutions that improve half-life, improve potency, improve breadth, and so forth. So we're moving up to something that basically is about 10-fold more potent and quite a bit broader. So this then became a clinical product. It's now in, I think, a bunch of different clinical trials. Before doing that, there were experiments with monkeys. So in these experiments, basically you take monkeys and you basically rectally challenge them with simianized HIV, which is a simian form of HIV. And then you see how much the antibodies will protect. So basically you inoculate with the antibodies and you later challenge them with HIV. So the protection you get with VRC01, the original antibody that Evelyn and people on his team discovered, you can see the kind of concentration needed here. With VRC07523LS, we see an improvement in potency of about 8 to 10-fold. So this was enough to go on to clinical trials. This is now in phase 2. This is the first clinical trial. We have results back from that. And there are about a whole bunch more, including several phase 2 trials. So one thing that's interesting is that a lot of this work relied heavily on computation and design. And it's a place where we can have, I think, a lot of impact as a community. And I think similar tools will be super useful for the coronavirus things that we want to do. Okay. I think I'm going to skip some of the algorithmic part, just because I want to go into detail here, but talk about our goals, but say that we need better algorithms, and we're going to talk about them to some extent. And we talked about provable algorithms already. And I want to just talk about the next phase of the class a little bit in the time we have remaining. So what happens next? So we're going to start by reading this paper, which I will present. Again, it will be a flipped class, so you will read the papers. There's a video to help you understand that. I don't want you to feel cheated that you're not getting a class. So I will be here next week for 2.5 hours, the whole time, talking to you about the paper, answering your questions, possibly going to the board and showing slides, motivated by your questions about this. But I think it's going to be more of a discussion and a more interesting class if we do it this way. And so that's why we're flipping that, but since we have these great resources of both the papers and the videos to do it. So we'll start by reading this and understand a little bit about how protein design works. This paper is paired by 2 chapters in the textbook, which I think will help you understand it and will help you read these things. But maybe you'd like to know some more about SARS-CoV-2 and more specifically, what these algorithms in structural biology might be able to do to help. I've hinted at that, but let me just talk a little more about it. So let's just take some pictures of how this looks. So we have, I mentioned, an RNA genome. It's wrapped up in proteins and lipids and so forth. There's spikes on the end, typical coronavirus. And one thing that happens, this is sort of wild type here, is that there can be mutations right, and deletions and insertions to this genome that happen with viruses. We want to understand what these mean because it's super important. This is a little bit historical because I'm going to go back to the beginning, but we'll get on to Omicron pretty soon. So what do these things mean? Well, one way we can understand that is by solving the structures. So here's structures of the spike protein, both in cartoon form on the left, space filling form on the right. It's trimeric protein, symmetric. So all the stuff you guys know about from engineering and science about symmetry are super relevant here. We'll understand what happens here with different mutations and variations and how these interact with partners. So it's similar exercise to what I told you about before. Get another picture of the trimer in cartoons from the top to look at this a little bit better. Let's look at some of the first mutations that arose, right? So we have these mutations. Let's look at some of them that occur here. Most famous is this N501Y mutation, asparagine to tyrosine. Looks like it increases the strength of binding in some sense, but you can also have deletions. These occur in the genome, but they also RNA genome, but they also have an effect on the overall shape here, geometric shape. So let's look at these in some sense. This is a location of this tyrosine mutation here, just in the structure of the spike protein modeled in as determined here. And of course, if we look at it from the top, we see that there are three because this is 3 copies of the same protein, all organized together like this. As we look at this, we can also solve structures of the receptor. This is the ACE2 receptor, basically it's the receptor binding domain of ACE2. We can see that in cases like this, there's an interaction. Probably this tyrosine is increasing the binding, which is what the virus wants to do. It wants to bind more tightly, so it has to have less virus in order to infect. So we can rationalize a direct meaning of this mutation. Now, of course, there have been lots of different variants since this initial picture. What do we know about Omicron? Here's some pictures that we'll talk about here. So there's a lot going on here. I'll let you kind of parse it, but one is this mutation comes up again and again. The combination of these different mutations basically seem to be a process of invading antibodies, which we're concerned about because we want the antibodies to do their job. So we're really interested in what these mutations do. I'll make a distinction here. What I'm showing here are visualizations of the mutations based on modeling. So we have structures that were not of the variant proteins, they're of the last variant in which we've modeled in the mutations. Now, most recently, we have actual structures of these and we can compare those. So if I say model, I mean we started with a structure and then we modeled in the mutations to try to figure out what they could do. Very powerful modeling, and I think it worked out quite well. So again, we could sort of try to visualize mutations even before solving the structures based on a model of the last variant or the progenitor variant and modeling these things in and trying to predict what the effect will be of these even before we have clinical data or even in vitro data. Again, we can compare different variants here. So we have Delta. We look at that mutation. See, Omicron has a lot more mutations here. We'll sort of talk about which of these are important, how they affect function, how they affect the ability of the virus to bind more tightly and to evade antibodies not bind to them. Again, this is a model again, by which I mean we started with a model of either Delta or wild type and we modeled these in. Beautiful pictures, by the way. I think a lot of people are drawn to structural biology by the sheer beauty of these intricate jewel-like proteins, even if they're doing terrible things. We'll also try to look at some mysteries. So for example, here is the lineage of the Omicron variant. Very interesting. It's shown in red down here like this, right? So what's really going on here? What can we piece together? So one theory, why such an extremely long branch without any branch? So one theory is that this circulated in a region with very poor surveillance. So we don't have a way of looking at that. A very popular theory is that this is continual evolution in an HIV affected individual. So basically, it's thought that there was a person in sub-Saharan Africa who was infected with HIV, but then went off the antiretroviral she was prescribed because the warlords would not let her get very close to the clinics she needed to get to. Terrible situation, of course, and we should be doing a lot more to make those drugs accessible, but because of that, the virus resurged and her immune system was to some extent compromised. At that point, she may have been infected with a version of COVID, which then took a long time for the immune system to suppress, and went through many variations and evolved Omicron, all in the same individual. What this really means in terms of public health is that we have to do not only a better job of getting vaccines to places that can't afford them as well, such as sub-Saharan Africa, but also getting them the infrastructure such as freezers and medical personnel, and also treat people with HIV infections consistently, because the perfect storm of having an HIV compromised individual in whom a novel coronavirus can mature and mutate is a situation dangerous not only to that individual, but to potentially the entire world. However, there are alternative theories here. A new paper that just came out, there's evidence for a mouse origin, that is a zoonotic transfer, probably from humans or another animal to mice, that something brewing up in mice, then reinfecting humans. So we don't really know, and this would be a really interesting topic to explore and inform with structural information. But we'll talk some about these in the form of the papers. Again, the first part I'm presenting is on basic tools, structural biology, algorithms, modeling, that can be applied really to lots of systems. Then with your papers that you're presenting, they either go into more tools and algorithms, which is great for someone who's a CS or algorithms person, or they could be something that's biologically focused on either machine learning or coronavirus. All right, what else will we look at here? What are some of these basic concepts we're talking about? Well, we're gonna talk more about these algorithms, from how to design algorithms work. Definitely going to try to, we talked about structures and rotamers a little bit. We'll talk more about configuration space, which comes from robotics, which is a great way to visualize these spaces. Again, here's the configuration space of a phenylalanine rotamer in some sense. So we'll talk about that. We'll also, I think I will skip a few slides here. We'll talk about configuration space a little bit later. Again, we sort of talked about this in the context of the isoleucine, in this case here. One thing I'll mention is that there's a thing we're trying to do, which is translate between biochemistry and computer science. They have different names for the same thing. So I had this slide before, but I want to mention this all has names that are very familiar to an engineer or computer scientist. So these different terms here have names in terms of, for example, CS230. So this algorithm here is basically a graph algorithm. And A star is basically a version of best first search. And these enormous streams of Boltzmann weighted energies, these are basically a infinite series or a stream. So the techniques we have developed in mathematics and computer scientists, even at a sophomore level class, can be extremely useful for shedding light on these things. And I want you guys to be able to communicate back and forth between these. Again, we will look at those kind of algorithms. We'll read this paper by Steve Mayo. We'll look more at the K-star approach to looking at protein binding affinity. Basically, the trick of this algorithm is that there are too many conformations to look at, but if you're looking at them in order, then you can get what is called in computer science an epsilon approximation algorithm. So these algorithms take an additional input from the user, which is an epsilon, which is desired accuracy. And they only have to look at a small, say, polynomial fraction of the input in order to get a good approximation. So we'll talk about those kind of algorithms and their utility instead. So again, I think the way to think about this is as follows. So we have the input, which is like structures, models, energy functions, all that stuff, algorithms to get an answer out. We always want to improve the model. What's the input? Structures, for example. We want to improve this. So for example, we might, in our first paper for next week, have the input be structures. We use an algorithm called dead-end elimination, which is a form of basically branch and bound. We might want to use these algorithms and we get out some kind of answer. But there are limitations to these algorithms. They might not have enough flexibility. They might not model ensembles. So what we'll do then is we'll kind of extend the input model to be more accurate, because we always want accuracy, better drugs that are more accurately designed. That then greatly increases the computational requirements, which is a real drag. So the job at that point for computer scientists and engineers is to come up with a more clever algorithm that can reduce the computational load, even given the new model. So then you might have an algorithm called minimized dead-end elimination that then looks at the more sophisticated input model and it's only a little bit more expensive, but gives you a much better answer. So again, each time what we're doing, now we add backbone flexibility, each time we make the input model more accurate, it blows up the computation to be something astronomical. But then, since we have the ability to do computer science and algorithms design, instead of just living with that, we're trying to buy more computers, which only has some limits. We can come up with better algorithms. The better algorithms process the information in a way that still can be done in a reasonable time. We get even better answers. So there's this continual interplay between improving the input model, blowing up the computation to be ridiculously expensive, and then clever algorithm design to still be able to do it in a reasonable amount of time. That's the reason that complexity matters in this sense. And we'll talk about that interplay in the class. So I think I'll skip this for now. We talked about provable algorithms. Again, a kind of thing that we like to read. This is one of my papers. We're not reading this except as background material for this class. But often we like things that combine experimental results with some characterization of the properties of the algorithm. So here's a case of designing a drug for combat cystic fibrosis. One of the things is we come up with these theorems or lemmas that are about the behavior of the algorithm. So it says that within the model that you're giving it, you have certain approximation and complexity guarantees. And this, of course, is then combined with actual results from structure, from ex vivo experiments, from binding. So the ideal story that we want, we'll get in every paper, sort of combines a characterization of how does the algorithm work and what can we say about it with an empirical characterization of the results. So we look for things like this when they're possible. And of course, in your projects, you might want to look for something that combines some aspect of this depending on your interests. Again, let's go back to some of the papers we might look at. We strongly believe that we can understand why things are neutralized by looking at the structures of antibodies binding to these spike proteins. So we'll read some papers on that. There might be some more recent ones since then, but that basically we can look at both the changes in shape and the changes in the interfaces here. And these are beautiful geometric objects to look at as well. For example, here, looking at the differences between these two proteins. You have wild type and we have delta, right? Delta variant. So this is a mutation from basically an aspartate to a glycine from here to here. And basically from the model that you can build, you can rationalize that you lose an interaction with the threonine on the partner, that is the thing it wants to bind to here on the ACE2 receptor, right? Basically, this is something that happens here, and therefore it loosens up the structure, makes it easier to do the transitions it needs to have function. So basically, once we have these structures, we can often understand the mutations that are happening and try to rationalize what's going on there. So we'll do some analyses like this, or those will be available as student papers to present. Again, we talked about using design to design therapeutics. We'll probably look at a few papers in that vein too. I already showed a slide based on this. We'll also look at some stuff you might not think about. So for example, what do other animals do? So animals can get affected with COVID as well. We worry about the cat family in particular, monkeys. Alpacas are amazing animals in terms of their immune system. So they make sort of Y-shaped antibodies like ours, but they also make another smaller form of antibody called nanobodies. These are single chain antibodies that are really marvelous. They make both kinds. They keep those beasts really healthy. This is basically camelids and sharks can make this. So camels, llamas, and sharks can make these. No other animal make these. So they raise these great antibodies against SARS-CoV-2. And there's the potential that we could take those antibodies, structurally characterize them as happened here, then humanize them, that is, change them so the immune system doesn't attack the antibodies, and use these as therapeutics. And one beautiful thing about these, we've worked with these in the past, is that they're much easier to make. They're stable at room temperature. You could transport them in trucks to places in deserts or jungles, and they still will be effective. So this is a really interesting therapeutic strategy. And again, we're interested in the structure of these things.
So is this just
Go ahead.
this is just like a random convergent evolution thing that sharks and camels randomly make this?
It's a really great point. It's a really good point. So I think that with the camelids, so that's alpacas, llamas, and camels, I believe that those are part of the same family tree, so they inherited it. We don't know why sharks have this. But I will tell you that people that work on this tend to work with the camelids instead of the sharks, because it's hard to have a farm of sharks that you're infecting with coronavirus and monitor them as easily, whereas the alpacas and llamas tend to be pretty docile. So most of the labs that do this have farms with alpacas. So I don't know why it is. I have looked into it a little bit. It's just a weird property, but no common ancestor that I know about. Great question. I'm happy to give you some reading on single chain antibodies and antibodies if you're interested. But it's a great therapeutic strategy.
That sounds super interesting. I never heard about this before.
Yeah, it's quite remarkable. We're doing it for HIV too. It's a great strategy. They're super stable. It's just a single chain. Also, they have it in you probably know that in antibodies have basically three really main loops that interact with things like the CDRH1, H2, and H2. So complementarity determining regions, heavy chain 1, heavy chain 2, heavy chain 3. And basically 2 and 3 are pretty short and even can be predicted computationally. 3 is quite long, so these long antibody loops. In nanobodies from the camelids, they can be really, really long, like 3 times as long, and they can go deep into enzyme sites to like disable stuff, and they're hard to predict the structures of. So they have lots of really amazing properties. So pretty interesting things to look at. And again, we're interested in a structural point of view. So we can talk more about that. I'd love to do that. Okay, I also want to talk about some recent algorithmic improvements. So basically, you might know that DeepMind, working on AlphaFold 2, has been able to predict structures of proteins really accurately. It's been a huge improvement in how they did this. So we're going to read some papers on how does deep learning enable end-to-end prediction of structures, right? This isn't protein folding, it's protein structure prediction, but it's really cool. And some papers will be available for you to report on this. I think they're really quite interesting. They beat the pants off every other method that does this prediction, including the highly developed Rosetta papers from David Baker's lab. It's really interesting stuff. So let's read some of those papers as well. We also want to read something about what's behind those, like why does this work? So basically it looks like end-to-end differentiable structures are really important for doing this. We might read some of these papers. We can also read outside the ones I'll suggest. We might look at some papers that looked again on deep learning for representing 3D structure. So we want not just to read the valedictory papers in Nature and Science, but the papers that talk about how do these things actually work. We'll also read some papers just on computational protein design, since we're designing assemblies. I don't know if we'll read this paper or something related, but I'm designing those papers now for you all to look at. And of course, there are some methods that basically mine the protein data bank to find ways to do design of different backbones in different cases. This looks like a really interesting and powerful technique. And there's several startups based on this idea now. So we're going to look at this technique, which is sometimes called MASTER or TERMS, are the two names for it, developed by Gavor Gregorian and Amy Keating and others. So I think we're going to read something in this vein too, or at least present them as choices. So a very useful technique for modeling the interplay between sequence and structure in order to do design. Finally, we want to have better ways to optimize these sort of K-star affinity algorithms. Great work from the great graphical models AI community on doing this recently. This would be a great paper to present for a computer scientist or engineer, and also previous backup papers here. These problems are related to not only computing the optimal solution, but counting the number of optimal solutions. Graphical model is basically a two-body energy function. We have self-energies and energies with one other's partner. So really interesting things, and of course, graphical models are used throughout computational biology. So those will be on offer as papers to read for someone interested in looking at the algorithms. And of course, the hope is that you can improve molecular design. Finally, we'll read probably some papers on how do we actually compute the structures from some experimental data. I know some of you with an imaging background might enjoy these papers. There's now QFIT 3. We'll look at the more modern paper that just came out. Also papers possibly on using cryo-EM, which is a really interesting technique for getting these structures. Because how do we look at the data and infer what the structures are? A really interesting problem. Particularly, how do we infer conformational ensembles? Here we want the conformational heterogeneity, the different shapes of these proteins as they vary under X-ray crystallography, NMR, or cryo-EM. So not just a single structure. Really interesting problem. Finally, coming to here, I want to say that before I showed you models of the Omicron variant, now my colleague Peter Kwong and his team at the Vaccine Research Center have a new structure just on preprint server looking at the Omicron variant, which is B.1.1.529, structural basis. What's going on here? What he finds is that a lot of antibodies don't work against Omicron, but some work really well still. So if we can focus on those, and if we can get people to elicit them, we're still in pretty good shape. So what's caught my eye here with this paper, which is one of the ones you all can sign up for, is what's the mechanism of neutralization? How does this actually work? Let's sort of zero in on this. So let's parse this here. Here the receptor binding domain. This is part of the ACE2 protein, which is on the human cell that gets infected all over the respiratory system, liver, and of course in the nose too. So basically, this is this guy here, right? This guy wants to bind, this ACE2, this receptor binding domain of the spike protein. So this guy wants to bind to ACE2. So here we have our spike mutation wanting to bind here, but the thing is the antibody, this particular one called, not a very good name in some sense, A1946-1, this binds here, causes what's thought to be a steric clash, like physically the molecules can't exist in the same place. So basically, if the antibody is binding to the receptor binding domain here, then it can't bind to the ACE2 receptor. So again, you kind of think of this as positive negative design in terms of what evolution is doing here. RBD, receptor binding domain of the S protein, wants to bind to the ACE2 receptor on the human cell, really wants to do this. Can he do it? Well, yeah, he gets there first. If the antibody binds to this guy first, then he blocks that binding. So interesting that they have a mechanism here and lots more data that combines the interface of sequence and structure. Here we can look at critical residues across different antibodies and rationalize how do these determine different potency, that is essentially binding affinity in cell potency here. So we'll look at some papers like this. I think they'll be really interesting. And the goal is here not just to look at the biology, but look at the algorithms that can give us better insight into these and to think about design and how we can design both better antibodies, better vaccines, better therapeutics. So a little picture of this. And again, sort of, this is a different topic. Again, sort of our goal for this, and certainly as a class and maybe in a project or papers, you'd like to have real biological insight. You like to characterize the algorithms, let's say how they will work beyond I ran it on this and it worked. You want to have some theorems or lemmas about the approximations, complexity, completeness, and so forth. It's ideal, and you want results that are basically about the in vitro, maybe even in vivo activity, and of course the structures you can compute. So I think that's an example of what we're looking for. We won't get it in every paper, we won't get it in every project, but it's nice to have some insight into those things. And indeed, in a laboratory, people are working on different fragments of these. So the mathematicians might be working over here, the structural biologists over here, and the folks doing in vitro studies in a different place, but they're all kind of combining this into one sort of vision. So how do I help with this? How do we help with this? Well, as I mentioned, the future presentations are going to be a little bit more didactic. I'm going to actually try to teach you some material, at least in the videos and so forth, that we'll discuss. Some classes will be pretty technical, but the recitations are really going to help. I think the exciting parts will be similar to what you saw here. And finally, I think the textbook will really help you. We're going to start with this paper next time, and I look forward to discussing it with you. Again, there's also a lecture as well as the material. Do try to read the textbooks. We'll read papers every week. The point is to read a lot. Keep in mind this view of kind of the dual nature. Structure determination is going from essentially gene to what is the structure, and structure-based design is going from the structure to how can I change, if you will, the gene so as to get different function. And that's how you optimize and design better therapeutics, or there's one way to do that. And finally, I want to mention that there's a whole science to how to determine structures better, not only in terms of the flexibility, but in terms of the mathematics you use to characterize symmetry. We might read some papers on this, but let me mention that it's sort of arbitrarily deep if you have a mathematical and computational bent. Happy to talk to you about that if you find the papers somewhat operational. We definitely can go off in the direction of papers that are really interesting from the mathematics of how these structures are constructed. Finally, we'll emphasize provable algorithms because I think they're the most useful ones in this field and because we can say the most about them, but we'll talk about the others as well. I think these are super useful in the course of an experiment that will give you sort of one pitch on why to do that is that essentially if we know the model is approximate, right? And if the design algorithm isn't exact or isn't optimal or isn't provable, then when your experiments don't match your predictions, you have two things that could be causing it, the input model or the algorithm. Which one do you choose? So classic form of optimizing experimental design is to take one variable out of the equation. So if we make it so the protein design algorithms are returning the best solutions in the model in order without gaps, then it turns out that when you have discrepancy between your predictions and your experiments, which you always will, that it's far easier to localize what you need to change in the input model because the algorithm is already giving you the best solutions in the model. And what that means in terms of running a laboratory or a class is that it means that students at an earlier stage of their career, knowing just some computer science and statistics, can make real contributions to the biological and biomedical goals. Whereas if you're simultaneously trying to figure out whether the problem was in the input model or whether it was the protein design algorithm, you really need to have essentially a PhD in computer science or statistics before you can even get going. So in my lab, it means that undergraduates and first-year students can contribute in really major ways to the process of designing and characterizing better therapeutics, because we know when we have a discrepancy between experiments and predictions, we can localize it to where the problem is in the input model. What I would say is, in measured terms, it makes the process of determining what needs to be changed in the input model substantially more straightforward. And that's the understatement of the year. So finally, please read this paper carefully. Look forward to discussing with you. And that's all I wanted to talk about in terms of the course. Okay, so we're near the end. I want to ask if there are any general questions, and then I will stay around for a few minutes in case anyone would like to talk to me. Consider it kind of an office hours. So any questions on what I have talked about so far or presented today? I want to turn off the spotlight here. How do I do that? Okay. Any questions? Yes, one question was slides. I will put a copy of these slides up today, and I'll do that sometime this week, and you can look at them again. All right, so since we're slightly over, what I'd like to do is I'll pause the recording, and I will